All right, and now let's continue further and take a look at the next part of the paper results. That's on the genetic characterization of the quote virus. So this section is still in the quote detection and isolation of a novel coronavirus results part of the paper, but it dealt specifically with genetic sequencing, so I'll cover this separately. So going further, so to further characterize the virus, de novo sequences of 2019 nCoV genome from clinical specimens, bronchoalveolar lavage fluid, and human airway epithelial cell virus isolates, presumably referring to the cell culture supernatant, were obtained by Illumina and nanopore sequencing. The novel coronavirus was identified from all 3 patients. So this de novo sequencing, and this de novo sequencing refers to genetic sequencing without using any any existing reference or template sequence, and the process involves DNA or RNA molecules being broken into smaller fragments, sequencing those fragments, and then a computer algorithm assembles the fragments into a quote complete genome sequence. All right, so that's basically when they're speaking about isolating the virus, what they're talking about is some next level genetic sequencing. And then the question is, well, what do they mean by this? So continuing further in the paper. So 2 nearly full-length coronavirus sequences were obtained from bronchoalveolar lavage fluid, and this is called BetaCoV, Wuhan, and so on, this identification. And that's this one, and there's the second one right here. And 1 full-length sequence was obtained from a quote virus isolated from a patient, and this is this one right here. This is the full length, the other one is partial length. But then they state that complete genome sequences should be 2 partial, 1 complete, because it says right here 2 nearly full. Yeah, so we have 2 nearly full and 1 full. Then 1 full length sequence were obtained. Then it says complete genome sequences should be 2 and not complete sequences of the 3 novel coronaviruses were submitted to GISAID, and it's this one right here they sent. 402119, that's this one. And they sent this one to the 20, this one right here, and there's the other one here, 40212. No, actually that's a different ID. So this one right here, let's see where this one is. Yeah, this one here. So 40212, that's 2121, that's this one. There's a 20, that's, they don't have that one there. Where's this one? This one right here is this. And going further, yeah, this one right here is the 22. And actually, I'll see that again. So this right here, I'm going to underline this one, is this. I just want to be double check, make it clear. And the next one is going to be this right here. Actually, this one right here. I just want to see the numbers. APSL. I'll underline this. Which ones match? And this one matches. And the next one is right here. But that's not matching. This one wasn't going to match. Yeah. That's this right here. Alright, so we got these and then a whole bunch of different lingo there. And have an 86.9% nucleotide sequence identity to a previously published bat SARS-like CoV, and that's this coronavirus genome. And the 3 2019 nCoV genomes clustered together with the sarbacovirus subgenus, which shows the typical beta coronavirus organization, and a 5-untranslated region, UTR, replicates complex ORF1AB, S gene, E gene, M gene, N gene, then R-UTR, that's the untranslated region, and several unidentified non-structural open reading frames. Alright, that's this ORF, open reading frame. So, I'm not going to go too much into what those all mean, but basically I'm just going to summarize that. So first of all, GISAID, or Global Initiative on Sharing All Influenza Data, is a worldwide database for genomic data on influenza, quote, viruses. And influenza, or, quote, the flu, is supposedly an infectious disease caused by, quote, influenza viruses. And the genetic organization mentioned over here So this, Phylogenetic organization. And then I have over here, so the genetic organization mentioned above are the supposed regions of the genome that are either non-coding or code for specific functions of the virus, such as replication or producing the spike protein. So this replicase, that's for replication, and so on. And that's the producing that enzyme, and so on. All right, so now continuing further in the sequencing part. So although 2019 nCoV is similar to some beta coronaviruses detected in bats, and that's figure 4, I'll go to that a bit, it is distinct from SARS-CoV and MERS-CoV. The 3 2019 nCoV coronaviruses from Wuhan, together with 2 bat-derived SARS-like strains, ZC45 and ZXC21, form a distinct clade. And SARS-CoV strains from humans and genetically similar SARS-like coronaviruses from bats collected from southwestern China formed another clade within the subgenus sarbecovirus. Since the sequence identity in conserved replicase domains ORF1AB is less than 90% between 2019 nCoV and other members of beta coronaviruses, the 2019 nCoV, the likely causative agent of the viral pneumonia in Wuhan, is a novel coronavirus belonging to the sarbecovirus subgenus of Coronaviridae family. Basically, they're saying these supposed conserved domains, or this is the replication domains, part of the gene of many of these coronaviruses. It's less than 90% of between the 2019 nCoV and other members of it. They're going to say that it's less than 90%, so they're going to call it a new virus. So since it's less than the 90% of that conserved region in many of these beta coronaviruses, they're going to call it a new beta coronavirus belonging to this other subgenus, and so on. Anyways, going further, so replicase domains ORF1AB are a set of conserved regions in the genome of the coronaviruses that encode for proteins involved in viral replication. All right, continuing further in the paper, and so here's how the whole genome setup looks like. So this is the BetaCoV. This is the 402119 with 29,892 base pairs. Where's that number? Here. This is right here. 402119. So that's the number 402119. And there's the base pairs all there. And there is the phylogenetic tree, and this is the beta one. This is the 2119 in red. So that's the one, and it forms it by itself. So it's different from all these other ones. And so there's a whole bunch of classification. And there's the MERS is over here. So MERS over there, and these are the SARS-CoV-1. Now these are the beta coronavirus, the new ones. It's going to be somewhere in there. Interesting. So figure 4, schematic of 2019 nCoV and phylogenetic analysis of 2019 nCoV and other beta coronavirus virus genomes. As shown are a schematic of 2019 nCoV, panel A, and full-length, this is panel A, this is right here, nCoV, and full-length phylogenetic analysis of 2019 nCoV and other beta coronavirus genomes in the orthocoronavirinae subfamily. Yes, very, very fascinating. So you have a whole bunch of these other these subtypes. So you have the sarbecovirus, sarbecovirus, and that's the beta one. The new SARS one goes inside there. There's the bad SARS like it, and there's a coronavirus one in there. So interesting. Note clade, or here it says note the clade with the beta COVs. Is this one right here? The supposed novel COVID virus, and you also have underlined bat SARS-like coronaviruses over here. So yes, it's similar to these bat-like coronaviruses. Anyways, going further, so summary of this genetic analysis part, or genetic characterization part. So summary, let's go over summary of the paper results section on genetic characterization of the virus. All right, and here's change the title name. So genetic sequencing of the clinical lung fluid samples, or just to make it more consistent, I'm just going to call that the airway fluid samples. That's the bronchoalveolar lavage fluid. And the cell culture supernatant obtained the virus genome. So they did genetic sequencing, and it was that de novo setup. And the virus genome was identified from all 3 patients. and 2 nearly full-length quote virus sequences were obtained from the lung fluid samples. 1 full-length quote virus sequence was obtained from the cell culture supernatant. And the 3 quote virus genome sequences were sent to the GISAID Global Flu Virus Database, and the genome sequences had an 86.9% nucleotide sequence identity to a previously published bat coronavirus genome. The 3 quote virus genomes form a distinct clade or grouping with less than 90% similarity of genetic sequences genes that code for proteins involved in quote viral replication. Yes, very interesting. So the main replication gene, it has 90% similarity, and then that's why it's going to be calling it a new coronavirus. It's not matching up with the other ones.